Welcome back, everyone. This is my final lecture for you guys. This is all about oxygen. Some of this stuff is review, but I'm going to introduce the concept of oxygen. I think everybody knows what oxygen is, but the majority of the discussion here will be on reactive oxygen species, what they are, how they're formed, and what are the good and bad things that reactive oxygen species can do, and how does our body defend against them. Obviously, we know that oxygen is essential for all anaerobic or all aerobic forms of life. Shown are just some of the things that oxygen can do for us. But again, pretty obvious here. The oxygen at high temperatures can be highly reactive in combustion reactions, but lucky for us, and at body temperatures, it's relatively inert. There's a high activation energy for oxidation reactions. The greatest use of oxygen in our body is committed to oxidative phosphorylation. I spent a lecture on that, being able to make ATP using oxidative phosphorylation is where most of oxygen is used in our body. In contrast, enzymes that use oxygen for hydroxylation, for example, use about 10% of the oxygen we consume, and less than 1% are converted to reactive oxygen species. So a very small amount of the percentage of oxygen, but important nonetheless, as we'll talk about today. Again, oxygen is usually inert. It requires high heat activation or high activation energy. At body temperature, oxygen can be activated by some metal ions such as iron, copper, and manganese. These redox active ions are kept in very low concentration in vivo, so they don't activate all the oxygen in our body. Okay, let's talk a little bit about the structure of oxygen and reactive oxygen species. Reactive oxygen species are just simply partially reduced reactive forms of oxygen. So we think about oxygen all the way here on the left, and water, it's being reduced all the way to water all the way on the right. The first reduction product is the anion radical superoxide, shown here. This is in equilibrium with the weak acid hydroperoxyl radical, shown here. Reduction of superoxide yields hydrogen peroxide, shown here, and then reduction of hydrogen peroxide causes a hemolytic cleavage reaction that releases the hydroxyl radical and the hydroxyl ion, shown here. And then again, water is the complete end product of O2. I don't expect you guys to know the structures or where all the electrons and things go, but I do want you to be familiar with the important reactive oxygen species, which are shown here on the slide, but I'll focus on the main ones here in the coming slides. Before we get to that, just talking about reactive oxygen and oxidative stress. Oxidative stress occurs when the rate of reactive oxygen generation exceeds the ability to neutralize them. This can result in an increase in oxidative damage to biomolecules such as proteins, lipids, and DNA, which we'll talk about. But reactive oxygen species can also be used by immune cells to destroy pathogens. So it's an equilibrium because we use reactive oxygen species in a good way to attack things like pathogens. It's also a feature of inflammatory diseases such as arthritis and diabetes. But an increase in oxidative stress and thus increasing the rate of reactive oxygen species can be due to increase in metals, overload and inflammation, increase in some enzymes, increase in alcohol, smoking, those type of things. Whereas a decrease can be the result of antioxidant enzymes, superoxide dismutase, we'll talk about that, some small molecules such as vitamins A, C, and E, glutathione, uric acid, and some other small molecules as well as proteins. So again, here's the main reactive oxygen species. The most prevalent one is hydrogen peroxide, shown here. This is fairly stable unless it's reacted with a metal. The hydroxyl radical is less stable, but the most reactive and damaging species that's shown here. The superoxide anion has intermediate stability and can be either oxidizing or reducing. And some other ones are the hypochlorite ion and the hydroperoxyl radical. Radical, I've used the term radical, also called free radical. It's just basically a cluster of atoms where you have one unpaired electron, shown in red there. It's extremely unstable, and it quickly reacts with other molecules or radicals to achieve the stable configuration. These can be formed by three major mechanisms in vivo, either by reaction of oxygen with metal ions, such using either the Fenton and Haber-Weiss reactions, for example. Talk about that again here in a minute. Or as a side reaction of the mitochondrial electron transport chain, or by normal enzymatic reactions such as the formation of hydrogen peroxide by fatty acid oxidases in the peroxisome. Here's the Fenton reaction, which I alluded to. So hydrogen peroxide can react with iron to form hydroxyl radicals. The Haber-Weiss reaction, hydrogen peroxide can react again to form hydroxyl radicals as well. There's also some metal catalyzed versions of the Haber-Weiss reaction as shown here. Just to give you a flavor, no need to memorize these reactions. Again, as I mentioned, they come from in vivo sources, but they can also come from ex vivo sources. So the major cellular sources are from the mitochondrial electron transport chain, as I mentioned. There are some other in vivo sources, as I mentioned on the previous slides, but can also come from outside sources such as UV light, ionizing radiation, smoking, air pollution, and some of the in vivo sources are listed here as well. As I mentioned, activation of polymorphonuclear leukocytes during inflammation conditions can result in reactive oxygen species. So, for example, there's a peroxidase that converts H2O, hydrogen peroxide and chloride ions to hypochlorous acid. Damage can occur due to reactive oxygen species. One of the most sensitive components of our body that is sensitive to reactive oxygen species is the cell membrane. These cell membranes are rich in polyunsaturated fatty acids, as we've talked about, which readily react with reactive oxygen species, and I'll show you the reaction here in a minute. This damage to the cell membrane can lead to reduced integrity of the membrane, so ion gradients can get screwed up, they don't function efficiently. You can also get asymmetry of the phospholipid bilayer, and essentially your cells can apoptose because of this. Here's the reaction of reactive oxygen species with polyunsaturated fatty acids. Again, the steps of this are not terribly important, but essentially you can get the reactive oxygen species reacting with these polyunsaturated fatty acids and lead to advanced lipooxidation products such as those shown here. In addition to fatty acids, polyunsaturated fatty acids, the hydroxyl radical can react with DNA by damaging both purine and pyrimidine bases and also the deoxyribose backbone. One of the most extensively studied DNA lesions is the formation of 8-oxyguanine, or also 8-hydroxyguanine. Shown down here, 8-oxyguanine. This can lead to permanent modification of genetic material due to this oxidative damage. And so this can be the first step in mutagenesis, can lead to cancer, premature aging, those kinds of things. In addition to lipids and DNA, the side chains of amino acids can react with reactive oxygen species. As I mentioned, reactive oxygen species are also essential for the function of cells, so they have good roles. The cells of the thyroid gland, for example, make hydrogen peroxide in order to attach iodine atoms to thyroglobulin in the synthesis of thyroxine. And as I mentioned, macrophages and neutrophils generate reactive oxygen species to kill some types of bacteria during phagocytosis. So here's a diagram showing that. During phagocytosis, a cascade of reactions leads to generation of reactive oxygen species to kill invading organisms such as bacteria. You can also get hydrolytic enzymes that are released from lysosomes, shown here, to help in the degradation of the debris that occurs during this phagocytosis. Our body has to defend against reactive oxygen species. They do that through either preventative mechanisms, repair mechanisms, or physical defenses. So many antioxidant defenses are shown here. So cells have a variety of enzymatic defenses against harmful effects of reactive oxygen species. One of the main enzymes is superoxide dismutase, which converts two superoxide anions into hydrogen peroxide and oxygen, as well as catalase, which converts hydrogen peroxide to water and oxygen. There are also some small molecules that are antioxidants: vitamin A, C, and E, uric acid. So you'll hear antioxidants a lot on pharmacy shelves and GNC, that type of thing, to aid in getting rid of reactive oxygen species that can lead to harmful effects, as we mentioned, such as cancer, aging, some inflammatory diseases. Shown here is the reaction of superoxide dismutase and catalase. Again, the reaction is not as important. I'm not going to make you memorize the reaction. Just know that these enzymes are important for antioxidant defenses in our body. There's also some of the co-substrates that are used here are shown. But again, it's more important that you understand superoxide dismutase and catalase being important in antioxidant defenses. Here's the mechanism of vitamin C. Again, it's not important that you memorize the reaction here. Just know that vitamin C is involved in antioxidant defenses. It can react with these reactive oxygen species. As I mentioned, it can lead, reactive oxygen species can aid in the development of diseases such as due to inflammation, such as arthritis, diabetes. Can play a role in infection, or obviously a role in cancer due to mutagenesis of DNA. It can also play a role in aging. On the flip side, it can also be exploited for radiation type therapy to kill cancer cells, for example. All right, a very short lecture, as you can see, and the study guide is basically the same as the outline. There's only really going to be a couple questions that come from this at most. I want you to understand that 90% of our body's use of oxygen is for the oxidative phosphorylation. I spent the majority of the time talking about reactive oxygen species. I want you to understand at a high level what those are: hydrogen peroxide. The other ones I mentioned, just know them by name, not by structure. How they're formed in vivo as well as ex vivo. Just some examples of that. What damages can occur from reactive oxygen species, mainly by reacting with polyunsaturated fatty acids for cell wall damage. It can react with DNA to cause mutagenesis. It can react with amino acids to screw up protein production pathways, for example. But then the good things that reactive oxygen species can do help in our immune system, or help during phagocytosis. And then what defenses our body has against it, the couple of enzymes I mentioned, and the couple small molecules. So again, very short and sweet, just a couple points for you guys to remember here. Obviously this is the last lecture, so I wish you guys the best on the exam and the best in the future. And again, please reach out with any questions. Take care, guys.